It is my great pleasure to introduce our keynote speaker today, Professor Anshul Kundaja. He's the Assistant Professor of Genetics and Computer Science at Stanford University, and he will talk today about the interpretable deep learning oracles to decipher the human genome. Anshul, thank you so much for joining us tonight, and the floor is all yours.
Thank you so much, Monica and Sophia, and all of you for being present today. Pleasure to be here. That was a really great introduction from Sophia, so I can hopefully build on some of that. Let me just share my screen. Are you able to hear me properly? Okay, great. Yeah, so I'm going to talk to you today about how we can sort of take these black box models, deep learning models, and kind of turn them on their head. And I'll hopefully showcase to you that, they are not these uninterpretable, powerful predictors. They are powerful predictors, but I hope to show you that we can, in fact, extract some pretty exquisite biology from these completely agnostic models, which is, I think, in some cases, quite fascinating. So this is a relatively new work, at least in the academic setting of relatively new. We had this preprint out on bioRxiv for about 2 years now, and the paper just got accepted yesterday. So I'm pretty happy about that. Things are taking longer and longer to publish, but helps us make our papers better and better. So there's always an upside. All right, so let's jump right in. So as Sophia said, there's been sort of a revolution in the variety of high throughput assays you can use to profile molecular properties of the cell, including DNA. And NIH has funded several large consortia over the last maybe 2 decades, which produce effectively these kinds of teracubes of information. You got the genome on one axis, you got hundreds of biochemical measurements on the other axis, and all of these are measured genome-wide in hundreds of cell types, tissues, and now in single cells. So think of this axis as basically infinite in some case. This axis is pretty large. This axis is thankfully finite, 3 billion base pairs. But given this quantity of data, it's pretty ideal that we can push these through powerful machine learning models with the goal of trying to understand how all this information is encoded in a single genome, 3 billion letters. And so today I'm going to give you a flavor of how we can take these data sets, kind of design powerful models that are well suited to model this kind of data, and then try to build powerful interpretation engines to decipher patterns learned by these models. So the key question I'm going to focus on today is what I call regulatory syntax of DNA. So we have genes and we have regulatory elements that regulate gene activity. And as I showed you in the previous slide, and Sophia mentioned as well, 2 very common assays used to profile regulatory DNA are ChIP-seq experiments that measure binding profiles of individual transcription factors, as shown here. And then you have assays like ATAC-seq or DNA-seq that give you genome-wide maps of accessible chromatin regulatory DNA. And our goal is to really try to understand, how does DNA sequence give rise to these beautiful, exquisite profiles of regulatory activity in specific cell types and tissues. And I'm going to focus on transcription factors today a lot, proteins that bind DNA, that recognize specific sequences, often referred to as motifs. And traditionally, there has been a lot of work on motif discovery that is trying to identify sequence specificity of individual transcription factors. What are the sequences they recognize in the genome? How do they bind? But transcription factors typically tend to bind DNA in a highly cooperative fashion. That is, they influence each other's binding. And that happens through complex motif syntax. Similar to how we have syntax in language, in the genome, you have these motifs embedded in sequence. There are interesting rules of arrangement, composition, spacing, and orientation. And this aspect has been relatively underexplored. And also how syntax manifests into cooperative binding of proteins is also difficult to study. So that's the question we're going to answer today, sort of higher order logic of sequence, regulatory sequence. And as Sophia mentioned, the traditional approach to analyzing this data using a machine learning formulation is as follows. Let's say you're given a ChIP-seq or a DNA-seq experiment, you got the genome, you got 3 billion base pairs. You can bin the genome into little chunks, let's say 1,000 base pairs. And the experiment that you perform essentially provides you labels for each of these chunks of sequence. So these labels could be binary. For example, is the resequence active or inactive, bound or unbound? Or they could be continuous. They could measure like enrichment scores or read counts directly. And then you typically take these sequences, millions of sequences across the genome mapped to labels, and it fits into a nice machine learning supervised problem. Sequences are inputs, scalar outputs. So you can fit any kind of classification or regression model to solve this problem. And there's been a lot of work in this space using a variety of different classes of models. We're going to focus on neural networks for one main reason, not just because they're powerful predictors, but because unlike many other classes of machine learning models, neural networks are one of the few that can derive representations from inputs de novo, learn patterns, learn how to combine those patterns and transduce them into complex outputs. And so that's why we're interested in them, because given a DNA sequence, we would like to try to see how we can map them to some molecular readout. So the traditional approach has been to take DNA sequences and map them to, say, binary readouts. Is this sequence bound or not by a transcription factor? So I'm just going to give a really brief overview of how neural networks work in 2 slides. You typically take the DNA sequence and you represent it as a one-hot encoding. So 4 channels ACGT, 1 at a 0 at each position, corresponding to which nucleotide is present. So for example, C right here gives a 1 at this position. The basic unit of a neural network is an artificial neuron, which you can think of as a pattern detector. All it does is it takes a collection of inputs. In this case, this neuron is looking at 5 base pairs right here. It takes these 5 base pairs, does some computation on it, and produces an output. In this case, the neuron is looking at 5 base pairs, which in this particular encoding is 20 inputs, right? 5 bases by 4 nucleotides. And the simplest kind of function for a neuron would be a linear function that just takes a linear combination of these inputs with some weights, W, and some bias, B. In this case, we have 20 inputs. So we would have 20 corresponding weights for each input. Let's just assume I made up these weights. We're going to learn these weights, but let's just assume I made these up. I'll arrange them in the same order, 4 rows by 5 columns. And if I visualize these weights as a logo, you can see that the weights of a neuron essentially encode a motif, right? In this case, it's CGATA. So an artificial neuron in the context of a DNA sequence model essentially is a motif detector. So in this particular case, I can take this input sequence, this piece of the sequence, perform the linear combination, and I get an output Z, 11.6, which is high and positive, telling me that the sequence that the neuron is looking at, CGATA, is in fact a very good match to the pattern it's encoding. Now, you can take these linear outputs of these linear neurons and push them to nonlinearities. So one type of nonlinearity is a sigmoid or a logistic function, which squashes the linear output in the infinite domain to a finite domain between 0 and 1. So it's nice if you want to predict probabilities. The other kind of nonlinearity is a ReLU, rectified linear unit, which is just a thresholding function that takes the linear output. And then if the linear output is less than 0, it sets it to 0. If it's greater than 0, it keeps it as it is. Simple thresholding function. So going back to our problem, now we're given a sequence and we want to map it to a binary continuous output. How do we do that? A single neuron learns a single pattern, right? A single motif detector. Clearly, your sequence potentially has many, many different kinds of sequence patterns or motifs. So maybe what we'd want is a separate neuron on average that captures each of the patterns we expect to see in the model. So instead of having one neuron that operates on these 5 base pairs, we'll have hundreds of neurons, each of which we will hope will learn a different pattern. And if you are given a pattern or a motif, what you typically do is you take this motif and then you scan the sequence. So you can do the same thing with this bank of neurons. You can take the neurons and then you can just basically replicate them with a stride of 1 base pair. So you're just moving the bank of neurons 1 base pair at a time and scanning effectively the whole sequence with hundreds of patterns. This is operational known as a convolution. That's why these are called convolutional neural networks. Now you don't have to stop at this single sort of layer of pattern detection. You can take the outputs of this first layer and push it through a second layer and a third layer. And each of these layers is learning progressively more complex patterns, building on the patterns learned by the previous layers. The final layer of the neural network, right, you can see right here, is a simple linear regression or a logistic regression, except that it's operating on a very complex representation learned by the model over multiple layers of convolutional neurons. So basically a CNN, at least for binary or continuous outputs, is a fancy logistic regression or linear regression model that operates on complex representations learned de novo from sequence. You train this model by feeding it thousands or millions of examples, which correspond to pairs of sequences and their corresponding labels. And you update the weights of these neurons. You randomize, you initialize them with random weights, and then you train them using stochastic gradient descent and various other approaches. And the algorithms optimize the weights of all the neurons such that you minimize some loss function that compares the predictions of the model with the labels, observed labels. So that's how you train these models. And there have been, since 2015, there have been many such models trained, which Sophia mentioned, that fall into this sort of categorization. Given sequences, you map them to scalar outputs. The unfortunate thing is a lot of these models actually let go of a lot of interesting information in the patterns in the actual profiles that are measured by these experiments. So let me give you a few examples here. So if you look at a ChIP-seq experiment that measures binding of a protein, and if you look at the read profiles, the read coverage profiles at base resolution, you will notice that you actually see some really beautiful exquisite patterns right here. So you see that typically where the protein binds, you see accumulation of reads flanking the binding side on the positive strand to the left and on the negative strand to the right. This is a ChIP-EXO experiment, which is like ChIP-seq, but we use an exonuclease to digest the DNA fragments all the way to the binding site. So you get these really beautiful, sharp footprints right next to the binding site. Similarly, DNA-seq and ATAC-seq experiments, as the protein binds DNA, you get exquisite footprints of read coverage profiles at base resolution. Traditional models are collapsing all of this information into a scalar at some low resolution, 200 base pairs. They just count the number of reads or they binarize this data. You lose all this beautiful structural information about how the protein's binding DNA and how it's potentially giving rise to this sort of profile, base resolution profile. So what we did is we decided to try to push these models to the next level and really see if we can model the data at its most basic resolution and most basic property. So for this, we took ChIP-EXO data, ChIP-Nexus, which is another slightly different protocol, which improves the ChIP-EXO data quality. And we looked at 4 transcription factors: OCT4, SOX2, NANOG, and KLF. As you know, the 4 classic pluripotency transcription factors in mouse embryonic stem cells. This was data generated by my collaborator, Julia Zeitlinger. Really fantastic data. You can see at base resolution, at a single enhancer, you can pick up these really gorgeous footprints of binding of the 4 transcription factors. So we wanted to model this data at its most basic resolution. And so what we did is we adapted these models in very simple ways. Again, the architecture is not the most interesting part. There are some interesting parts, but this isn't the most interesting. All we did is we built a fully convolutional model. We take sequence, and instead of predicting a scalar, we map the sequence to base resolution count profiles on the positive and negative strand, okay, from the ChIP-EXO experiment. So this is kind of nice because we have to do minimal processing of the data. We just take the ChIP-EXO data, we map the reads, we filter the crap, and we get base resolution coverage of reads on the plus and minus strand, and that's it. You don't have to worry too much about peak calling and all that stuff. And so our goal is to take sequence and map it to these stranded base resolution profiles. And so for that, what we do is our model is essentially a convolutional neural network with a few bells and whistles. We use dilated convolutions just so we can expand our receptive field with minimal, without exploding the parameter space. And we use residual connections just because it speeds up learning and allows for deeper models. There were a few important things we included. As you know, all these experiments have biases. If you fit a powerful model to this data, the model is definitely going to learn the biases. So what we do is we not only give sequences an input, but we also give a bias track. The bias track is typically, for example, it could be a patch cap bias for ChIP-EXO. It could be an input wild type control bias for ChIP-seq. It could be DNase bias or TN5 transposase bias for DNA-seq or TAC-seq. It's just a bias track that captures the inherent bias of the enzymatic assay or the procedure itself. So that goes in as an input. And in the final layer, the model automatically regresses out the contributions of the bias track. Okay, so essentially the model is an additive function in the last layer of the sequence component, which is this residual convolutional neural network, and a bias track. Okay, so the regression is learned during training. There's 2 other changes we made. We decided to model the data at 2 different scales. So we're looking at a 1,000 base pair sequence, and we wanted to model the total counts that fall in the 1,000 base pair sequence, which captures in some sense the occupancy of that region. But we also wanted to model precisely how those total counts are distributed at each base in the sequence. And if you think about it, a probability distribution that captures that really nicely is a multinomial, right? At each position, you have a probability of observing certain reads. You have a total number of reads that you throw at the sequence. And those total number of reads get distributed at each position as a function of that multinomial probability. So we have 2 loss functions operating. One is a loss function on the total counts, which is a mean squared error on the log of the total counts. And the other is this multinomial negative log likelihood, which really captures the distribution of the reads at each base pair, or how those total counts are distributed across the positions. And we also multitask these models across the 4 transcription factors. So we simultaneously learn joint models across the 4 TFs. So how do these models actually perform? They perform really quite well. These are 2 examples. These are 2 enhancers. The way we evaluate the model is through cross-validation. We hold out some chromosomes, we train on a few, we do this in 10 folds. So basically, we are predicting on entire chromosomes not seen in training. And as you can see, the observed and predicted tracks at single base pair resolution are often remarkably similar, kind of undistinguishable in many cases. The multinomial model also, as I'll show you in the last few slides, also has an inherent denoising effect because you're learning these probabilities per base. And if you have low coverage of a region, you will often observe zeros, right? But those are not real zeros. They're just observed zeros because you didn't have enough coverage. The multinomial can effectively denoise and desparsify the data. And so you can often get really nice predictions of expected signal at every position in the sequence. So those are two like random examples I picked. If you do genome-wide evaluation, you actually see that the models do as well as replicate experiments. So just to unpack this plot, what we are showing here is, let's say this is the observed track. We can mark the positions where you see these spikes, the footprints, right? These are the ground truth positions. And then your goal is to see how well you can predict those ground truth positions in the predicted profile. That's one metric. We have several metrics. You can also compute like the Jensen-Shannon divergence between the two profiles, or you can compute a negative log likelihood based on the model's loss. In all cases, whatever metric we use, we see that in this case, we're looking at the area under the precision recall curve, which captures the precision of positional prediction of these spikes in the profiles, the footprints, relative to the ground truth. And so the black curve is if you just took random profiles, that's the like sort of the lower bound, that's completely randomized profiles. As you can see, it's quite terrible. The green curve is the area under the precision recall curve. If you just took the average of the observed profiles. So if you took all the observed profiles, you compute an average and just use the average to predict all the profiles, what would be the performance? The yellow is if you use one replicate to predict the other. So how well do two replicates concur in terms of prediction of the footprints? And blue is the model. So in all four cases, you see we are either slightly better or slightly worse than replicate concordance. So you can see chromosome-wide, the models are doing really well at precisely detecting positions of footprints. And similarly, if we use continuous metrics or similarity, we get very similar results. What's very interesting though is this is sort of predicting how well the model does in predicting the shapes of the profiles. How does a model do in predicting the total counts? In this case, the model is not as good as replicates. It's still state of the art. It's very high. You can see the correlation between the observed counts and the predicted total counts is in the range of 0.6 to 0.65. But the replicates are extremely similar, 0.96. So what's going on here? Basically, what we're seeing is the effect of how much you can pull out of local sequence. So remember, we're trying to model these profiles using 1,000 base pairs of sequence. And what the model is showing us is that 1,000 base pairs of local sequence can almost perfectly predict the shapes of the profiles. But the total occupancy is not just a function of the local sequence, but is a function of many other properties that are not captured by the local sequence. That includes distal interactions. So there are clear examples of SNPs, genetic variants in distal enhancers affecting binding levels of transcription factors and other enhancers that are, in some cases kilobases or megabases away through long-range interactions. Similarly, chromatin state can override the impact of local sequence and affect total occupancy. So total occupancy is a more complicated measure to predict from sequence, and local sequence doesn't cut it all the way. It takes you pretty far, but not all the way. If you add additional covariates as inputs, like chromatin state, or you increase the length of the sequences, you start improving these performance metrics even more. Okay, so I'm going to switch gears. So that's just telling you that the models are actually quite powerful and predictive. That is, given new sequences in the same cell type that it has not seen before in training, it can do a really good job predicting the profiles, and to a large extent, the total occupancy as well. You may ask me, why are you trying to do prediction? when you already have the data, nobody's ever going to like give you half the chromosomes and ask you to predict on the other. Well, the reason is because, as you'll see later, we're going to use these as oracles. And like Sophia mentioned, if your model is able to predict profiles for unseen sequences with very high performance, even if it's in the same cell type, they need not be sequences in the genome. You could do perturbations to sequence in the genome and then see what happens, right? The model could predict what could happen. So that's why it's important to have a high prediction model. But now we'll look under the hood and see what the model is actually learning. So I'm going to switch gears into the interpretation piece. I'll pause for a moment and see if folks have one or two questions. I don't want to take too many questions, but if you have a burning question that will prevent you from understanding the rest of the talk, I'm happy to take one right now.
Great, thank you, Anshul. Does anyone have any question? Maybe the simplest way will be for you to speak up and ask your question.
I have a maybe naive question.
Yes.
So you're looking at the window of 1,000 base pairs. Why do you did you consider smaller windows, maybe like 100 base pairs or 200 base pairs? Because if I remember right, transcription factor binding sites are usually very narrow.
Mm-hmm.
So why the choice of the 1,000 base pairs?
Yeah, it's a very good question. The binding sites are in fact narrow. They're about 200 base pairs. If you're looking at ChIP-EXO, they're even narrower. They are like 40 base pairs. But as I'll show you, context really matters. And you can test this computationally. So you can just fit models with different sizes of sequences. And you see dramatic gains if you go from 100 base pairs to 2,000 base pairs. After 2,000 base pairs, the improvement is much, much minimal. So the bang for the buck isn't as much. But up to about 1,000 base pairs, 1,500 to 2,000 base pairs, you get actually very, very significant, and almost sometimes in some cases, twice the performance as you get from just local sequence. So there is influence of proximal context on binding. Even though the binding site is just 200 base pairs, what's happening in the local context actually has a significant impact. And I'll show you some examples at the end where we do simulations of what happens when you insert a genetic variant and changes allele. How does the model respond? And you'll see some really interesting blast radii of variants. Okay.
Thank you. That's very helpful.
Yeah, so let's now get into model interpretation. So let's say I have this enhancer in the genome, the sequence, and the model makes this really accurate prediction of this specific profile. The question you might ask is, how is it doing that? And what are the important nucleotides in the sequence? So the variety of different methods to do this. We have one called DeepLift. It's basically a backpropagation method that takes the output of predicted output of the model and then recursively decomposes that output into the contributions of individual neurons in each layer. Okay, you backpropagate this through the model recursively until you reach the each nucleotide. So basically think of it like all the neurons in the model voting on the contribution of every nucleotide to the predicted output. And so you get these really nice base resolution contribution scores of individual nucleotides to the predicted profile output. So what does this look like? So we can take an actual enhancer right here. This is the OCT4 gene. This is a very famous distal enhancer, very well validated. This is a known enhancer which has been knocked out and it has an effect on OCT4. So a lot of biology is known about this enhancer. The other reason this enhancer is interesting is because it's also an auto-regulatory enhancer. You have OCT4 binding that enhancer and regulating OCT4. More so, all 4 transcription factors in fact bind that enhancer. So it's a complex binding landscape. And what we can do is take the model, and for the same sequence, we can use DeepLift to obtain the contribution scores of individual nucleotides with respect to each transcription factor. So you can see each TF has a different footprint, and the same sequence is giving rise to these different footprints for the 4 different TFs. And so you can see that they look kind of similar, but there are subtle differences. You can see one motif right here, like lighting up for all 4, but you can see this piece right here, TGAT, lighting up specifically for NANOG, and then these GGGG sequences, this one as well, really lighting up much more strongly for KLF4. And if you just map these kind of high importance or high contribution subsequences to known motifs, you can basically get very high resolution annotations of predictive motif instances showing complex, intricate interplay between different motifs. So we can use this to kind of like annotate any enhancer in the genome in terms of which pieces of it, of the sequence, are being utilized to give rise to different binding profiles, or different transcription factors are co-binding the same enhancer. But that's assuming you know the motifs. Clearly, the neural network is somehow learning these motif representations de novo. So can we actually reverse engineer the motifs themselves instead of just like interpreting individual sequences? Can we kind of consolidate these important high contributing sequences across the entire genome and relearn predictive motif models? And so that's what we do. The way we do that is through a simple approach conceptually. We take every enhancer or every binding site for each transcription factor, and we use DeepLift to obtain contribution scores. So imagine you have 3 sequences bound by the TF. These are the made up DeepLift scores. We then throw away pieces of the sequence that are deemed not important, and we identify these contiguous regions of high importance, which we call seclets. And then we cluster these based on their similarity, and then we collapse them into non-redundant motif representation. So each base here is capturing the average contribution of every nucleotide, or each of the 4 bases at each position. Okay, so it's kind of like a position weight matrix, but it's not. It's not capturing the frequency of nucleotides at each position. It's capturing the average contribution score of each nucleotide at each position in the motif. And this subtle difference will be important as we go ahead. So we call these contribution weight matrices, similar to position weight matrices. So what does this look like? We looked at 4 transcription factors, right? And if you look up these 4 transcription factors in a motif database, what you will see is typically, maybe 4 or 5 or 7 motifs. Often they will be kind of similar. You won't exactly know why they're different, slightly different versions of these motifs across the board. What we find is that you actually need about 50 different motifs to explain binding for these 4 transcription factors. So you may ask, well, you're just making up 50 motifs, about, how do you know they're actually different? The nice advantage we have of the CHIP EXO data is we not only have a motif, but we have a corresponding footprint corresponding to that motif. So for each of our motifs, which are aggregates of these seclets across the genome, we can compute aggregate footprints for each of the 4 transcription factors. You see really interesting patterns pop up. So these are the position frequency matrices. These are the corresponding contribution weight matrices. And what this matrix tells you is which transcription factors are the motifs important for, right? So some motifs like OX-SOX, the OX-SOX heterodimer, are important for OX-SOX and NANOG binding, but not KLF4. Other motifs like these 2 are very important for NANOG, but not so much for any of the others. You can see some motifs like this one being very important for KLF4, not the others. So we're seeing kind of interesting combinatorial interplay of motifs across different transcription factors. You'll also notice another interesting pattern if you look closely enough. Direct bound motifs tend to have really sharp footprints, as you can see right here. So this is a NANOG motif to which NANOG binds. You can see very sharp footprints. But NANOG also binds to indirectly, is tethered or recruited to, as you can see right here, ZIC3, SOX2 motifs, and others. And you'll see that the footprints tend to be more diffuse. So whenever a TF is recruited through indirect tethering, you get a diffuse footprint, and when it binds directly, you get very sharp footprints. And this is true across all the TFs. We can also precisely count how many motifs on average are found, a distribution of predictive motif instances in any enhancer. And you can see that they usually range between 3 and 4. In some cases, you have 8 instances. You can also basically get a notion of how many predictive motif instances you have across the whole genome for these transcription factors. So as I mentioned, there is evidence for tethered binding. The model not only learns the direct bound motifs, but also learns indirect binding through tethering. And one such example is ZIC3. And so we did an experiment for ChIP-Nexus for ZIG3, and in fact found really nice sharp footprints for ZIG3 at these precise locations where NANOG binds ZIG3 indirectly. Okay, so again, you can see the difference between the sharp footprints and the diffuse footprints. So there's lots of interesting evidence of tethered recruitment of TFs by motifs of other TFs. We also find, I told you we found 50 motifs for 4 TFs. So there are many interesting variations of these motifs, and of course we can't validate all of them yet. We genuinely believe many of them are in fact distinct binding modes, but one instance where we had clear proof was, or relatively clear proof was for NANOG. So for NANOG, we learn actually 3 distinct versions of the motifs. They have a shared TCA component right here, but they have slightly different sort of flanking bases. So this one is a primarily TCA motif. This one has TCA with this TT-AT sequence, and this one has a GG-ATT. What's very interesting is each of these 3 motifs have been previously validated in the literature, either using crystal structure. So for example, this TA-AT piece of the motif is in extremely close contact with the NANOG protein in a monomer crystal structure of NANOG with DNA. The GG-AT sequence has been previously also validated in vitro experiments, and the TCA component of the motif is reasonably well known. So we have evidence that, at least evidence, I guess, I wouldn't say it's 100% proof that these different versions of the motifs are in fact bound by NANOG, and they give rise to distinct footprints, which have evidence for, in fact, being in contact with the protein in crystal structure. So that's sort of on the motif discovery piece. So I'll jump to syntax now. And as I mentioned before, we wanted to go beyond these motifs and motif instances. I just wanted to show you that even in the space of motifs, we do pick up interesting new properties of motifs if you derive them directly from the model. But let's jump to syntax and see what happens. So there have been many hypotheses about motif syntax and how they operate in regulatory DNA. One is this idea of very strict spacing. It's almost like a grammar, right? So imagine you have one motif and another motif, and they are always separated exactly by 6 base pairs, 10 base pairs. So we wanted to test how often we see this in the model. So what we did is we took all pairs of motifs and we just computed the distribution of distances between them, predictive motif instances. And we looked at motifs that had very high frequencies of specific distances. Okay, so on the x-axis is the distance, on the y-axis is the frequency of a pair of motifs separated by that distance. And you can see these ones in blue are the ones that really have skyrocketingly high frequencies, much more than just expected by chance. So what are these? So we noticed that there are 2 types. The first is what we call composite motifs, which are pairs of motifs that in fact form protein-protein interaction direct complexes that bind DNA. So like OX-OX NANOG, right? OCT and SOX form a heterodimer. They often form another a complex with NANOG. And when those 3 TFs or 2 TFs bind together, they often show very fixed spacing. But then you also see a bunch of these motifs which have like pretty large scale fixed spacing, like SOX2 and ZIG3, exactly 46 base pairs. This one is exactly 68 base pairs, 62. So this would be quite interesting if it were real. But we noticed that across the board, every time we saw these what we call like long range or distant composite motifs, they always fell into retrotransposons. Okay, so basically these are just repeat elements. You see many instances of these in the mouse genome. And as many of you might know, enhancers actually evolved a lot from these retrotransposons. So the retrotransposons are effectively like, harboring evolutionary banks for evolution of enhancers. And these transcription factors actually bind many of these classes of retro elements extensively. And so what's happening here is basically this fixed spacing is not necessarily in fact a property of some grammar or something like that. it is in fact an artifact of the background sequence that the TF is binding in. It's just an artifact of the TF binding an exact repeat sequence, a very similar repeat sequence that is replicated, 60 times, 100 times, 400 times in the genome. And what's beautiful is the model actually learns to discount that. So if you just look at a position frequency matrix of these motifs, these long motifs that we find, you basically pick up the whole repeat, because as I said, the same sequence is present multiple times, right? But if you look at the importance scores, if you look at the contribution weight matrix, the model beautifully ignores parts of the repeat and focuses very specifically on nucleotides that the TF binds to. And you might say, well, it's looking at the footprints, so obviously it's learning that. These are test sequences. It has not seen the footprints. It's predicting the footprints, and then from the predicted profiles, you are inferring the nucleotide contributions. And so you can see in this retrotransposon, OCT4 binds these nucleotides, SOX2 binds these nucleotides, NANOG binds this connection, right? In a different retrotransposon, you again have differential usage of the retrotransposon sequence. Different parts of it are being used by different TFs to bind DNA. You can imagine how these retrotransposons then evolved into specific enhancers for specific TFs by losing all the background sequence based on simple evolutionary constraints. So that's pretty cool. Now we can take repeat elements, retrotransposons, and we can zoom into functional nucleotide sequences. And we have another project, which I don't have time to talk about today, where we've actually been able to take really large families of retrotransposons and look at zinc finger transcription factors, which is one of the largest classes of transcription factors that bind many of these viral elements, trying to repress them. And we can actually learn arms race relationships between evolution of the retrotransposon sequences and the binding affinities of these transcription factors through these kinds of models. So that's one aspect of syntax. To summarize this piece, we do not see any evidence of strict motif spacing in the mouse genome for these 4 transcription factors, except for direct protein-protein complexes, which we call short-range composite motifs, and artifacts of binding in retrotransposons. So we believe that syntax is loose; it's not fixed. There is little evidence for really strict syntax. But as I'll show you, there is actually very strong evidence for soft syntax. So here's one example of soft syntax. And also I'll show you again the difference between a position frequency matrix and a contribution weight matrix. So if you look at the NANOG motif, the first one I'd shown you, there's this TCA component, right? The TCA and this little piece of sequence on the left. If you look at the position frequency matrix and look at the flanking nucleotides, yeah, it looks pretty dead. But if you look at the contribution weight matrix, you see a fascinating pattern. You see this TAAT sequence popping up at regular intervals around the motif. And in fact, if you plot this as a heat map, each row here corresponds to a different instance of this motif, TCA, and the heat map is capturing the contribution scores. You actually see these really nice bands, right? Almost exponentially decaying periodic contribution scores. And here's a visualization of that. And in fact, if you just compute the Fourier spectrum of this pattern, the frequency that really lights up is 10.5, which is the helical turn of DNA. So there's something to do with NANOG that has this interesting property of it having a preference where it binds DNA in some kind of periodic fashion. And what's pretty cool is for several of these types of homeodomain factors like NANOG, there is evidence from in vitro NCAP-SELEX data that homeodomain factors like NANOG, this has never been shown before for NANOG, but for other homeodomain factors, they can form homodimers and they can bind DNA in this sort of homodynamic fashion through 10.5 base pair periodic sites. Okay, so that's potentially what's happening here. What's also really fascinating is that 10.5 base pair periodic patterns are observed for motifs only in the context of NANOG. So if you look at the OXOX motif, in the context of OCT4's binding, you do not see the 10.5 base pair periodicity. But the same motif if you're looking at it in the context of nanog binding sites, you see a very strong 10.5 base pair periodicity. So it's something very specific to nanog. It's not some artifact of the chip exodata. So we can go in the genome and actually see what's happening. If our hypothesis is that this is happening because nanog preferentially binds with itself or with other transcription factors at intervals of helical turns, then we should see this in motif spacing in the genome. And that's actually, in fact, exactly what we see. So if we take our motif instances from our model, from the CWM, and we just graph the positional distribution of pairs of nanog motifs, you see this beautiful 10.5 base pair periodicity, right? If you use position weight matrices without using the model, just you scan the sequence with the motifs, and you don't use the predictive importance, that pattern disappears. So it's not easy to find. It's basically you need the model to really highlight these instances, predictive instances, and then you really see it light up. It's not just nanog and nanog. Yeah, did I have a question? Okay, so it's not just pairs of nanog homodimers. You also see the same relationship for SOX2 and nanog, OCT4, SOX2 and nanog, nanog and ZIC3. So nanog with any other transcription factor that it cooperates with appears to have this 10.5 base pair periodic preference, soft preference for spacing between the motifs. Now you might say maybe this is an artifact learned by the model. Well, we can go back to the actual data and see if we see that in the profiles. How is the model able to learn it? This is the actual read counts. So we've simply aligned the motifs, we've aligned at one nanog motif, and we're looking at read counts at nanog motifs separated by various distances. And you can see in the reads itself, you have this really nice periodic pattern. Now you see it because we've done the alignment, but if you actually just visualize the data, it's very hard to see it. So it's not kind of making this up. It's actually able to learn it from the subtle pattern it sees in the reads, and then it identifies a periodic spacing of these nanog motifs, which are in sync with these sort of periodic amplification of the read distributions, and that's how it's able to learn the patterns. So that's talking about, so I showed you examples of soft syntax, which is, and the model sort of learning helical periodicity de novo from the sequence as a function of these base resolution profiles. How does syntax actually affect TF cooperativity? That's a different question. So the syntax could exist in the genome, but does it affect binding in different ways? That's the big question of one TF versus the other. So we built basically two kind of oracle experiments mimicking what you would do in the lab. One is synthetic sequences. So this is more like think of it like a MPRA, a massively parallel reporter experiment, where I can synthesize DNA, right? I can insert motifs. I can create whatever syntax I want, and I can shove it into the cell, and I can measure what happens, right? I'm going to do this kind of in silico. So I'm going to synthesize sequences. I'm going to insert some motifs. I'm going to change their spacing and orientation, and the model is going to tell me what's going to happen. Okay. I can also do equivalent of like in silico CRISPR, where now I'm not taking synthetic sequences. I'm taking genomic sequences. I know that this sequence has a bunch of motifs as predicted by the model. And now I can systematically mutate motifs and look at the effects that the model predicts would have on binding. So we do both of these. So this is an example of a simulation, the synthetic simulation. This is a synthetic sequence. I placed nanog motif at this position. I placed the OXOX motif at this position. Now I started moving this motif towards the nanog motif. And what you're seeing is the model's predictions at base resolution, how nanog's binding profile is changing as the distance between these motifs change, and how OCT4's binding profile is changing as the distance between the motifs change. And then I can just plot basically the change in the binding footprint for nanog, which is the gray signal, sorry, the gold signal, and the corresponding change in the OCT4 binding signal as a function of the distance between the two motifs. And you see something really cool. You see that OCT4's binding profile doesn't change at all. It doesn't care where nanog is. It can be 100 base pairs away, it can be 5 base pairs away. It really doesn't care. Classic pioneer factor, right? It binds its motif and it doesn't care who else is around it. Nanog, on the other hand, is really getting pushed around. As OXOX moves towards NANOG, you can see NANOG responding very strongly, almost like an exponential rise. And it's not just an exponential rise, but you also get this really beautiful 10.5 base pair periodic frequency. So every time the OXOX motif is in multiples of 10.5 base pairs away from the NANOG motif, you get a cooperative effect. You get a strong rise in binding. And you can see that this cooperativity actually extends up to exactly 150 base pairs. If you plot further, it rapidly dies down to basically baseline, which is exactly a nucleosome turn, right? A nucleosome is about 150 base pairs of DNA. And so you're seeing sort of this short range cooperativity of OXOX and NANOG operating through potentially nucleosomal nucleosome distance through a helical cooperative effect. Okay, and this is again being learned de novo from sequence. There's no information that has been given to the model. It's just picking this up. You just have to query it to figure it out. We can do the same analysis in the genome. So this is the in silico CRISPR experiment. So this is the same OCT4 enhancer I showed you before, where OCT4 and NANOG bind. And you can see the model's prediction here. Here's the OXOX motif, here's the NANOG motif. So let's delete the OXOX motif and see what happens. You can see the OCT4 sequence dies completely, the NANOG sequence, sorry, the OCT4 profile dies completely, the NANOG profile dies completely. So if you delete the OXOX motif, NANOG cannot bind either. It doesn't just destroy OCT4 binding, it also destroys NANOG binding. On the other hand, if I destroy the NANOG motif, OCT4 doesn't care, but NANOG's profile gets hit pretty bad, although it does get partially recruited by OCT4, tethered binding. And if I graph this plot across all the enhancers in the genome, I actually see a very similar plot to what I saw in the synthetic experiment. Basically, again, you see OCT4 not caring for NANOG until it comes really close by. But NANOG really, again, has this exponential rise in cooperative binding with a 10.5 base pair periodic signal. The signal is weaker, obviously, because there's more heterogeneity in vivo sequences, which you can control for in the synthetic sequence, but you can clearly see the pattern. And what's really interesting is we can do this for all pairs of motifs and TFs. And across the board, we pick up really interesting cooperative effects. Some are pretty long range, up to 150 base pairs. Others are pretty short range, like NANOG with NANOG itself is a shorter range, like 50 base pair cooperativity. Similarly, SOX2 with SOX2. KLF4 kind of doesn't care about anybody. So we can essentially get these really nice asymmetric cooperativity plots of how motifs of one transcription factor affect binding of another at different distance ranges. So this is short range protein-protein interaction cooperativity. You can see the OXOX motif influencing everybody, NANOG kind of responding to everybody except KLF4. And you also have nucleosome range cooperativity by OXOX, not so much by others. So I made up a lot of stories. How do we know this is actually real? So why not do the actual CRISPR experiment? We did in silico CRISPR, let's do in vivo CRISPR. Julia's lab was kind enough to do these experiments, beautiful experiments. So let me show you a few examples. So here's an enhancer that has NANOG and SOX2 binding it. This is what the model predicts. The model predicts this profile for SOX2. It also predicts that this motif is really important. It's the one driving the binding of SOX2. So what we do is we take this motif and we mutate the TT. This is what the observed data actually looks like. So it's pretty nice. You can see very nice concordance at base resolution between the predicted profiles and the observed profiles. Now we take this motif and we mutate two base pairs, A and G. Why did we pick these two nucleotides? Because the model told us that these two nucleotides are the most important. If you break these, you're going to break everything. So we make that mutation and we propagate it through the model, and the model predicts this profile. It predicts that the binding profile at this position gets attenuated very strongly. And there's also partial attenuation at another SOX2 binding site, which is a few hundred base pairs away, or 50, 60 base pairs away. If you perform the actual experiment, you see something very similar. You can see the binding of the footprint of SOX2 completely abrogated at this position and attenuation at the distance. And this is exactly like the SOX2 double cooperativity that you see for short range up to 50 base pairs. Now we can do the same thing for nanog. So the same enhancer, we're going to measure nanog's binding. So here what you see is, again, the predictions of the model and the observed data for both mutants. We are mutating the SOX2 motif. You can see it actually influences nanog binding quite strongly. Not just at the site, but across the board. There's attenuation across the board. We can reverse this and now take the nanog motif and again perform a similar mutation experiment. And here you again see if you mutate this nanog motif, you disrupt a nanog footprint at this position with some abrogation further away, not too much, not too very far. So there isn't a long range effect as predicted by the model. It's very short range cooperativity. And in fact, if you mutate the nanog motif and you measure SOX2 binding, the model predicts no effect and the data says no effect. So we're getting really nice high resolution predictions through single nucleotide mutations or motifs. And the model is not only able to predict the profiles accurately, but also the range of effects, the short range, medium range, nucleosome range effects. How much time do I have, Monica?
I think you're still good, but we probably want to move to the discussion part soon.
Okay, yeah, I have 2 more slides left.
Perfect.
So this was sort of direct validation on binding. This is what the model is trained on. Can the model generalize to other readouts? That's an interesting question because it's trained on binding. Is binding relevant? Like, does the syntax have any relevance to other measures of regulatory activity like chromatin accessibility or gene expression? So we can test that. What we do is we take the representation of the model, the final layer, we freeze it, and we fit very simple linear models to that final layer to ataxic data. So it's a linear model that's fitted on the sequence representation learned by the binding model. Okay, so we're testing whether the representation learned by the binding model transfers to a chromatin accessibility through a very simple linear transformation. Okay. So there are 2 ways to test this. One is if we just take the motif instances learned by the model and we use them as features, how well does it predict differential accessibility after knockdown of OCT4 or SOX2? And you can see the model does quite poorly. This is a very simple additive model. It's assuming that the output, the differential effects after TF knockdown are entirely a function of the motifs in an additive fashion. You can see the correlation is pretty bad. If instead you take the representation of the model, the final layer, which includes the cooperativity and the syntax and all of the nonlinear effects, you actually get really nice correlations. So what this tells us is that whatever higher order syntax that the model is learning in its representation through a very simple linear transformation can very accurately fit to ataxic data after TF knockdown. And again, this is on held out experiments, held out sequences. So there's no training test contamination. We can do the same thing for MPRAs. So we can take enhancers and we can measure expression levels in reporters. And we can again take the model's output, the binding layer, the last convolutional layer, the sequence representation, and we can fit a simple linear model to that and see if the syntax representation again transfers to expression. In fact, it does. So what this is telling us that the binding representation the model is learning is quite universal. Of course, the readouts are different, but it's a very simple linear transformation that makes a binding readout into a accessibility readout into an expression readout. So I'll just summarize there. I have 2 more slides. I just want to show you a quick preview of variant effect prediction. This is very fresh off the oven analysis, but it's quite exciting. So I just want to give you a quick preview. But before I do that, I just wanted to summarize the talk. So I showed you these new BPNet models that can map raw DNA sequence to base resolution profiles at a really high performance accuracy. In this case, on par with replicate experiments. I showed you interpretation frameworks can allow us to discover motifs, motif instances, syntax, cooperativity, using sort of these in silico oracle like experiments. And the model's predictions we showed were validated by CRISPR motif perturbation experiments, differential chromatin accessibility after TF knockdown, reporter expression. And we've now extended these models to ChIP-seq, cut and run, DNA-seq, ATAC-seq, single cell ATAC-seq, PRO-seq. The model works out of the box pretty much. There's very few changes. You just need to usually play with the capacity of the model a little bit, and it works out great. So I want to show you really quick, one quick example of how in the future we could use these models to do really cool things like take a genetic variant and predict the effects of different alleles on multiple layers of readouts. So here I'm showing you, this is a validated SNP that affects binding of SPY1. Okay, so it's called a SPY1 binding QTL. The SNP is right in the center right here. And the model is predicting that for the C allele, you get kind of weak binding. Here's the DNA signal, and here's H3K27 acylation signal. These are 3 BPNet models. One is trained on ChIP-seq, the other is trained on DNA-seq, the other is trained on histone ChIP-seq. Okay, and you can see they're trained on different sequence sizes. This one is 1 KB, this one is 1 KB, this one is 6 KB. This is what the observed data looks like. You can see how noisy it is. And the model does a beautiful job denoising and desparsifying the data. What we can then do is swap the allele of that variant from C to a G. And you see across the board, you see a big jump in signal, right? You see the binding signal rise, you see the DNA signal rise, and you see the acylation signal rise. We can compute the difference between the two alleles at the profile level and obtain what we call the blast radius of a variant. So that means if you make a single nucleotide change in the variant, up to what distance do you actually see an effect on multiple readouts? ChIP-seq, DNA-seq, of the TF and the histone marks. And you can see really amazing, like you can see actually there's a nucleosome periodicity the model has learned. It's weak, but it's there. So you have different blast radiuses of the same variant on different types of regulatory readouts. And then if you use DeepLift to interpret these sequences for the 2 different alleles, you get remarkably similar predictions. These are 3 completely independent models. What it predicts is the reference allele, you're zooming into 200 base pairs right here. There are 2 SPY1 motifs, one right here and one right here. I'll just go back. When the allele is a C at this position, this SPY1 motif is destroyed. And there's a weak SPY1 motif that gives rise to this weak binding profile, weak DNA signal, and weak acetylation. When the C switches to a G, you make a SPY1 motif, which is very strong. It activates, and you have a strong cooperative effect that drives binding, drives accessibility, and drives histone modifications. So we are able to now do this for hundreds and thousands of variants in hundreds of cell types. Able to make these extremely high resolution predictions of nucleotide effects of single nucleotide variants cooperatively between transcription factors, and really understand how the sequence code of these regulatory elements is transduced into these different kinds of regulatory readouts. And so I'll just stop there and thank people who really pioneered some of this work. Ziga was the first author of this BPNet paper. He's the one who built the architecture. Avanti built all the interpretation tools. Julia's lab did amazing ChIP-XO experiments and CRISPR experiments, and Amar helped a lot with some of the coding as well. And these are my funding sources. Thanks.
Thank you so much, Anshul. That was an amazing talk, and we have already many questions in the chat. So I'll jump right into that. I promise Damien will return to his question. I think it pertains to the initial part of your talk. Mm-hmm. Would using NGS whole genome sequencing data be useful in this context, using sparse model to make genetic causal inference? GWAS seems underpowered for rare variants network effect, however. Is there machine learning that seems promising?
Yeah, absolutely. You know, we have been trying to work on the variant scoring problem for years. And there have been papers published in this space. In fact, some of the first applications of deep learning models were for variant scoring, the DeepSeek model, the Bassett model, and so forth. We tried to stay away from that because we realized that the models that we were training at that time were extremely unstable. And you can try this for yourself. you can take some of these binary models, the coarse resolution models. You can train 3 or 4 different versions of them, just random seeds, random folds, and then make predictions. They are all over the place. So we worked for many years to stabilize the models, and there are 2 ways in which you can stabilize. One is by avoiding the summarization, because you can get from a sequence to a summary scalar in many different ways. The models can learn many different strategies to predict a label. So you get the same prediction, but you get completely different interpretations of how you're getting those predictions. By making the model predict profiles at base resolution, you constrain that space. It doesn't have all the freedom in the world to just predict total counts. It has to also predict this base resolution profile. So that actually constrains the models and makes them significantly more stable. In fact, we have a paper that, in fact, has an approach that makes this even more stable. We add some additional priors in the training phase to dramatically stabilize the models even more. Now we are in a situation where we believe the models a lot more, and we can obtain these really stable and high accuracy inferences of variant effects. And so we do actually have really nice results already on GWASs for many different diseases by training models on very interesting cell types, microglia for Alzheimer's, coronary smooth muscle cells for coronary artery disease. And the models do an amazing job fine mapping hundreds of GWAS loci. It's actually quite cool, because you don't need massive cohorts of individuals with case control studies. You just need like one great experiment in one great cell type that's causal, and the models can go pretty far.
Great, thank you. Another question we have is, what was the sequence composition of the spacer in the synthetic experiment? Do you think it is also context dependent?
Yes, in the synthetic experiment, actually, I kind of cheated. We run this experiment over tens of thousands of backgrounds to avoid having a context effect. You're absolutely right that if your context is off, you will have some interaction effect between the motifs and the context. And so if you want to really get extremely accurate predictions of the interactions specifically between the 2 motifs, we synthesize many benign background sequences as predicted by the model. We can push these backgrounds to the model. If the model predicts a flat line, we say, okay, good, that's a benign background. We generate tens of thousands of these benign backgrounds. We insert the motifs, we space them, and then we average the predictions over all of them. So we are marginalizing the effect of the background effectively.
Thank you. Another question is, what sequences would be treated as out of distribution samples for BPNet? Is there any benchmark to show how BPNet generalize for out of distribution?
That's such a great question. I was hoping somebody would answer that. So I use the word oracle a lot. These are restricted oracles. They can, to some extent, interpolate. They definitely cannot extrapolate. So for example, if I dumped some random sequence motif into this model, it would make some random prediction, right? It's only as good as what it's trained on. So there are 2 ways in which we make sure we're not out of distribution. The first is any rules we learn on synthetic sequences, we try to replicate on genomic sequences. If the 2 agree, then we're good. If they don't agree, we need to break the tie through some other mechanism, like an actual experiment that we need to actually validate which of them is telling the truth. So in some sense, the synthetic experiments and the genomic deletion experiments allow us to work in a space that is reasonably constrained. The other way we try to be in distribution is for any property we're interested in, we plot the distribution of the property in the genome, and then we make sure that our simulations are in that realm. We are hoping to build better methods to test out of distribution effects, but I think your question is bang on spot, and it's a great one, that these models are not actual oracles. They cannot tell you what's going to happen in situations that they will never train on.
And so I see that we have one more question. We're also on time. Anshul, are you okay with taking one more question?
Yeah, sure.
So we have a question. Do you have any thoughts on how to incorporate variation located more than 1 kilobase from the motif, say in a distal enhancer?
Yes. So I think in the preview that I showed you, get to that. Yeah. So for these ones, right? The histone model, for example, actually is using, in this case, a 6 kb sequence. But our latest models, we've been able to go to 100 kilobases. And we're continuing to expand the receptive field. We think in a month or two, we'll be at a megabase. We might even be able to do whole chromosome. And the trick is not to go whole convolutional. It's not going to work. The way to think about it is as you increase the distance, the genome behaves more like a graph and less like a sequence. It is locally a sequence, but it's globally a graph. So it is kind of a little bit pointless to model megabases of sequence when you have small chunks of that megabase, in fact, driving signal, right? So what we're doing is we're building local models of 10 kb or so, right? Which we can stride at single base pair resolution. And then we identify loci locally that have strong local effects, right? Like we see it idea. And they become active nodes in our graphs. Again, then we can propagate information across nodes through a graph model. And that will allow us to generalize to arbitrary distances without worrying too much about the size of the sequence. So we'll get to megabase scale models pretty soon. We're already at about 100 kilobases, and we can take super enhancers like what I showed you. Look at blast radii. You know, if a super enhancer has like 3 or 4 individual DNA peaks in them, we can break one at a time. We can break pairs of them, and we can test for interesting distributed cooperativity, not within an enhancer like I showed you, but between enhancers. And we have really nice evidence that actually works quite nicely. For example, for CTCF binding sites, we have simulations where we have disrupted a particular motif, and you again see this blast radius kind of emanating out, you know, up to 20 nucleosomes or so on either side. So you can look at these, you can actually pick up long range effects of mutations through these kinds of models as well.
Thank you, that was great. I see that we are on time, so I think we might want to stop here. I would be open to letting people discuss a little bit longer, but I don't know if everyone is available. So let me maybe right now just extend my thanks to our great speakers today, Sophia and Anshul. Thank you so much, that was amazing. I also want to thank our breakout room facilitators and thank you all for attending. And if you would like to spend a couple more minutes chatting, please feel free to stay online.